Let's run through a demo analysis of caloric restriction, an intervention that extends the length and health of life in various species. First, we need to find some studies to include from GEO. We limit the search to series of type expression profiling by array, which is just microarrays. Once you find a study to include, copy its accession to your analysis. Next, we need to add contrasts. A contrast is just a control and test group that will be compared to each other. Select the study that we just added, then add control group samples. You can use Shift plus click to select multiple samples at once. Then add the test group samples. You can also add more than one contrast per study. As a shortcut, after selecting samples, you don't have to click the input box. Just start typing and press enter to add the group. If you make a mistake or decide not to include a particular contrast, just delete that contrast. If you're ever stuck, click the info circle on each page for detailed instructions. You can optionally specify a source for each contrast. A separate meta-analysis will be performed for contrasts with the same source. A meta-analysis that includes all contrasts is always run. That's it. Click Analyze Genes to run the analysis. This can take 30 seconds or so. The 50 most significant genes are plotted. The x-axis labels are links to the gene card's entry for that gene. The data for all genes can also be downloaded. We can also use the expression signatures for each source to search over 26,000 studies for similar and opposing contrasts. This takes about a minute. The first two results, CMAT2 and L1000, are large drug expression studies. After these are the top 30 most correlated studies. The table shows the predicted sample groups for a study and the correlations between our search signature and the contrasts with that predicted group in it. In this case, the most correlated study is also on caloric restriction, so let's add it to our analysis. We would also need to add contrasts and sources, just like for our first study. You can also add analyses that others have contributed and contribute your own analyses for other people to use. Please also check out the docs for details on how meta-analysis improves search results and how our gene expression search engine compares to others. Thanks for watching.